Hello, everyone, and welcome to today's webinar, Mass Photometry versus Macro Mass Photometry, Advancing Data-Driven Decisions in Drug Development. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Refine. To learn more, visit refine.com. We encourage you to participate today by submitting any questions you may have. To do so, simply type them into the Ask a Question box and click Submit. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now introduce our speakers for today's event. Jacintha Victoria, Market Development Manager at Refine, and Salma Jalal, Market Development Manager at Refine. Salma, I will now pass it over to you for your portion of the presentation.
Thank you, Cassie, and thank you everyone for joining us online today. Jacintha and I are from the market development team at Refine, and between us, we serve scientists in the cell and gene therapy and protein-based therapies markets, respectively. I've been with Refine for about 3 years now, and one of my favorite parts of my job is getting to know our customers and finding out what their analytical challenges are, and then seeing if we can bring value together to see if we can simplify and accelerate their workflows through mass photometry technologies. In this presentation, I'll start by explaining the principles behind mass photometry and macro mass photometry before highlighting some applications of mass photometry in critical quality attribute analysis for antibodies and in the mRNA therapeutic space. Then, Jacintha will take over and walk us through how mass photometry is being used for assessing AAV purity, and finally, how macro mass photometry can be applied to the characterization of larger viral vectors. So let's start off by taking a step back and looking at why we need to characterize biological drugs in the first place. Critical quality attributes, or CQAs, are defined by regulatory agencies and are essential to monitor throughout the drug development process, as they help ensure that the final product will be of high quality, safe, effective, and compliant with regulatory expectations. From previous experiences and industry learnings, we have seen that failure to adequately manage and monitor critical quality attributes can lead to serious consequences, including clinical trial failures and, in some cases, safety issues that could result in patient harm or even death. Given how important it is to monitor critical quality attributes, there are well-established platform technologies that are widely used for CQA assessment of traditional drug modalities, like proteins and monoclonal antibodies. However, it can be challenging to adapt these platform methods to newer trending modalities like bispecifics, ADCs, mRNAs, and viral vectors. For instance, high sample demands associated with traditional technologies can be particularly challenging when working with novel modalities that have higher costs of production compared to traditional monoclonal antibody formats. Or in mRNA analysis, where capillary electrophoresis and HPLC methods that are typically run in denaturing conditions, which involves altering sample properties before analysis. So you might not have an idea on the true purity profiles of your molecules from these analytical methods. Or in the case of AAV analysis, in this space, AUC methods are typically used for empty full analysis, but AUC methods are slow and complex and typically require a subject matter expert or outsourcing, which can then delay project timelines by days to weeks. And if you're not familiar with mass photometry, let me introduce this technology to you, as it can overcome some of these challenges. As it measures the mass of particles in solution using nanograms of sample in minutes. Mass photometry is a microscopy-based technique that measures the interference between light scattered by molecules near a slide surface and light reflected from the surface. So if you look at the first panel, you can see that we're shining a light into a sample, a sample of molecules in solution. And then in the next panel, you can see that when the molecules are near the glass slide, they scatter light And the signal that we're looking at is the interference between the scattered light and the light reflected back by the glass surface. And that signal is called a contrast signal. So that interference signal that we measure is called a contrast signal. And you can see there, each of those little contrast dots actually corresponds to a single particle event. So a single particle coming close to the surface and moving away from the surface. And then what we can do is do a calibration measurement. So you just measure a calibrant before you measure your samples that you're interested in, and we can convert that contrast signal into a mass signal as the contrast signal scales linearly with the mass of the molecules. And we can just perform a calibration step so that you can then get this output of a mass histogram that's composed of the readout of thousands of single particle mass measurements. Since we're working with a readout as generic as mass, you can look at just about anything that affects the mass distribution. So for instance, you can look at impurities like aggregates that are larger than your desired monomer population, or you can look at particle binding like antigens to antibodies that would then result in a mass shift. I've given you protein examples, but mass photometry can measure any biomolecule that falls in the mass range of 30 kilodaltons up to 5 megadaltons. So this could include anything like proteins, antibodies, small viral vectors like AAVs, DNA, or mRNA. And in the context of mRNA, the size range in that case would range from around 200 bases up to at least 10,000 bases and slightly higher. Furthermore, mass photometry is an easy-to-use technique that doesn't require a subject matter expert. And I love this video because it shows you that. If you can see here, this is all that is required when starting a mass photometry measurement. The hardest part of doing a measurement is pipetting onto the system. So over here, you're pipetting 10 microliters of sample onto the system, and you're using a typically 10 nanomolar concentration of sample. So that's going to equal nanograms of sample for a typical antibody format. And that was the most difficult step, because once you close up the lid, then you go onto the software and click acquire, and the software handles everything else for you. Analysis is happening in the background as the movie is being acquired, and then at the end you get this readout again for the measurement. And as you can see there, it's a benchtop instrument, so it doesn't take up that much space. And yes, it's very easy to use. So anyone can do it after half a day of training. And then as we said, mass photometry is for the mass range of 30 kilodaltons up to about 5 megadaltons. But what if you want to work with larger particles? That's where macro mass photometry is going to come in. So if you want to work with particles that are larger than 40 nanometers up to around 150 nanometers, you're going to be using macro mass photometry. So what is that? Macro mass photometry is a technology that is building up on mass photometry by adding an additional layer of information. So here, not only are you getting information on a measurement of contrast, which is a proxy for mass, but you're also getting information on the particle size or diameter. So you're doing a typical mass photometry measurement of looking at the interferometric scattering, but now you're doing it with a Z sweep. So you're going to have let me go back. So you can see, I'm going to use I think I can use a laser pointer. Let's do this. So you can see over here, you're going to add a Z sweep into there to scan through, and then you're also going to get a readout of the particle size. And then this is very nice for being able to distinguish between different populations of particles that are perhaps of the same mass. So they'll have a similar contrast measurement, but then are actually of different sizes. So you'll have a smaller particle in diameter here that can be differentiated from a larger in diameter particle here, even though they have a similar contrast measurement. Or in a similar idea, you can look at a particle that has two different particles that have a similar size, but have different masses, and then differentiate based on these two parameters of information that you have in there now. And then on this slide, we have an overview summary of the different applications for mass photometry and macro mass photometry. In case you're familiar with the product lines, this also gives you an idea of how they map onto the mass photometry. We've got two products out, the 2MP and the SAMX. The 2MP will typically be used for molecules like antibodies or mRNA, and you'll be using the SAMX if you're only doing AAV anti-partial full analysis. And then we have the Caritro MP, which is working on the macro mass photometry principles that can be applied for non-enveloped and enveloped vectors, and to be looking at the purity there. And then I'm going to kick us off with an application focus now, and looking at how some of our users have used mass photometry for characterizing attributes associated with antibodies, in particular for monoclonals, bispecifics, and ADCs, and how they've used mass photometry for this. So I'm going to be showing a few examples of how you can look at antibody aggregation, as well as looking at target binding stoichiometry as well. So this slide helps demonstrate how MP is also antibody modality agnostic, and it doesn't require any method development steps when you're moving from working with a monoclonal format to a bispecific or a ADC format. You're able to characterize a range of different formats in minutes using nanograms a sample without any method development. Because of MP's wide buffer compatibility and the measurements being column-free, we only require one universal optimization step when you do a measurement. All you have to do is dilute onto the instrument. And as you can see, all of these samples, monoclonals, bispecifics of different size, and ADCs, have been measured on a 2MP using the same methodology, so just being diluted in PBS. And then for these measurements over here, we collaborated with Absolute Antibody. They're a developer who produce antibodies of lots of different formats. Here we're showing you some monoclonals. And then in their work, they're providing purity measurements to their customers on all the antibodies that they produce with a platform SEC method. But sometimes their platform SEC method returns inconclusive results, and method development is required for optimal measurements for some of their situations, like here. Here we're going to show you a side-by-side comparison of their SEC runs with the MP measurements of certain samples. And then these representative examples show cases where in A, you have the SEC method, the platform method working well, and this method works well for most of their antibodies, but they do have situations like in situation B and situation C, where in situation B, they have some molecules that behave differently in the SEC columns and have delayed retention times. So they're just slightly shifted. So there's something going on, but it's not terrible enough that you won't get any information out of it. So you can still get a little bit of an idea on the purity. But as you can see in the case of situation C, they have some molecules that behave so strangely on their SEC columns that they're unable to get any meaningful data out. And like I said at the beginning of the slide, all of these formats are monoclonal antibodies. So you're seeing these differences in retention behaviors even between antibodies of the same format. And this isn't even considering additional complexity that comes when you're working with bispecifics or ADCs. And in these three situations, MP was able to quantify aggregation when SEC could, and also when SEC could not, or was unclear. And even in the case where SEC did work and gave you some information, mass photometry was able to give you baseline resolution for the monomer and dimer population. Over here in panel A at the bottom, it gave you baseline resolution on the monomer and dimer, and also could detect this additional 40 kilodalton fragment peak that SEC could not detect. And then finally, you're getting comparable measurements. So MP can be used as an orthogonal to SEC, and it's giving you measurements 20 times faster than SEC can, and using 100 times less samples. So here, you're using nanograms of sample with a 1-minute measurement versus you needing micrograms and a 15 to 20 minute runtime for SEC. And then this is an example where we're considering the use of analytical ultracentrifugation as a orthogonal to SEC. So typically when a candidate moves to later stages of development in preparation for regulatory dossier submissions, a column interaction-free method needs to be used for antibody aggregation assessment. Typically, AUC is used for this, and it often has to be outsourced because these instruments are so complicated to use, and typically you don't have a subject matter expert in-house to run these measurements. And typically with this outsourcing, it can cost thousands of dollars and delay project timelines by days at least, or up to weeks when you're waiting for results to come back. So here we show mass photometry as an orthogonal to, and showed it benchmarked to AUC for assessing antibody aggregation in a forced degradation setting. So we have here a NIST antibody standard that has been exposed to a heat stress. So you can see the control condition in blue and the heat stress condition in orange. And you can see in the mass photometry measurement on the left, you had this superior resolution to the AUC measurement where you're able to resolve 4 aggregate species, the dimer, trimer, tetramer, and pentamer, whereas in the AUC, you can only resolve the dimer with baseline resolution. And then for the resolvable peaks, MP measurements were comparable to the dimer and trimer aggregation levels that were detected by AUC. So you can see that on the graph on the right. And doing a scaling for the difference in the measurements being so a dimer and a trimer measurement. And then also just the ease of use for these measurements as well. Mass photometry is a 1 minute measurement versus AUC, which is a 6-hour measurement. And then that just also means that you're able to run multiple replicates on MP if you wanted to, which is typically not done with AUC because of the expense and the amount of sample that is needed. So you're going to need to use like 8,000 times more sample also for AUC. So there is just the comparison of a 30 nanogram measurement for mass photometry measurement versus the 250 micrograms that you need on AUC. So mass photometry is a ideal column interaction for orthogonal to SEC that is returning results in 1 minute with nanograms of sample again. And then moving into another application for mass photometry in the antibody space is looking at studying binding stoichiometry and whether you have binding in your samples. So this is more from our collaboration with Absolute Antibody, where we're showing how mass photometry can be used to rapidly identify antibodies that bind their antigen with high affinity. So Absolute Antibody, as I said before, produce a range of different antibody formats. So they not only work with monoclonals, but also produce bispecifics in other formats. And with bispecifics, they bind to their targets with a different mechanism. So they're binding multiple targets. And over here, we're showing a panel of bispecific antibodies that were tested for their ability to bind HER2. So they either had a 1-to-1 or a 1-to-2 or a 1-to-1 or a 1-to-2 stoichiometry with the HER2 antibody. And then as you can see here, they either had a 1-to-1 or a 1-to-2, or here was only a 1-to-1 because this arm is binding a different antigen, or 1-to-2, and then that arm would be binding a different antigen there. And then here, this bispecific is only a 1-to-1 binding. So you could check that you had the right binding stoichiometry in these measurements, in these samples with a 1 minute measurement using nanograms of sample. So as long as the affinity is in the nanomolar range with these nanomolar measurements, you're able to rapidly identify high affinity binders and bind and confirm their binding stoichiometry in minutes with nanograms of sample. And then in this data set, working with one of our customers, we were provided with a panel of therapeutic antibodies that had either low, medium, or high affinity binding to EGFR. And this classification was based on BLI data from the industrial partner. But we actually did this study initially blinded. So Refine didn't know which antibodies were low, medium, and high binders before reporting the results to the collaborator. And then once we did the mass photometry measurements, we could show the antigen antibody binding stoichiometry, and we were able to provide separate affinity estimates for each binding stoichiometry for 1:1 versus 1:2, and that these measurements correlated with the BLI data. So now I'm going to move into looking at some examples of how mass photometry can support mRNA development and attribute measurement for mRNA, like integrity, purity, and stability. Over here, we're showing you how mass photometry is able to measure mRNA of different sizes over a wide size range. So this is a panel of mRNA of therapeutically relevant sizes from 1 to 4.5 kilobases, as well as a 10 kilobase saRNA. And we want to highlight here that all of the samples were measured in the same conditions, and the measurements of the length actually all had a relative error of less than 5% over this really wide size range. And you're able to measure self-amplifying RNA with the same methodology that you're using for your smaller mRNA therapeutics without needing any method development steps. It's just diluting onto the system. And then coming back to the idea of measuring the true identity, the true purity profile of your mRNA. Here we show how most methods used for mRNA purity assessment that involve capillary electrophoresis or HPLC methods are typically run in reducing or denaturing conditions. So this means that they will not be able to detect non-covalent oligomeric mRNA species. So in this example, we have a commercially available mRNA EGFP that was measured by MP by mass photometry in native conditions and in denaturing conditions. And you can see on the left in the denaturing conditions, the purity that was measured of around 80% correlates or agrees well with the purity provided by the manufacturer in the certificate of analysis from capillary electrophoresis data. But actually, when you then look at this, when you look at the sample in non-denaturing conditions, where non-covalent interactions are intact, you're able to see these dimer and trimer populations that are in the sample as well, that are not apparent in techniques that are run in denaturing conditions. And again, this is a 1-minute measurement using nanograms of sample versus like a 15 to 20-minute run that you would need for CE or HPLC methods, and micrograms that you would need for those methods as well. In this next example, this is a study published last year by a group at Genentech, where they compared a range of techniques for mRNA impurity profiling and showed that mass photometry is an effective stability indicating method. In this figure, mRNA transcripts with and without poly-A tails were subjected to heat stress. So you can see on top in the control, the untailed and the tailed transcript had a size length difference of around 100 nucleotides, which corresponds to the presence of the poly-A tail in the poly-A tailed samples. And then the authors have also noted that the purity levels that were measured by MP were aligned with data that was obtained by IP, RPLC, and CGE. And then commented on how mass photometry is able to reveal the presence of these mRNA dimers for both samples. And then the mRNA dimers were considered non-covalent forms of non-covalent dimers since they were absent in the IP, RPLC, and CGE profiles. Furthermore, they wanted to just test that further and make sure that these dimers were non-covalent. So they did a heat stress where they where they kept the samples at 37 degrees for about 5 days, and then they could see that the dimer populations were reduced, and you got an increase in shorter transcripts of these pre-peak areas, like shorter transcripts, smaller than the monomer. And then finally, in this sample, in this figure that we've selected from this study that was published earlier this year from Sanofi, from a group in Sanofi, they showed that mass photometry is able to rapidly characterize mRNA samples in process as well. They tested the robustness of MP to measure crude mid-process samples from varying stages of the development process workflow. We don't know exactly what was in the matrix buffer, but they've been taken from different stages, so they have differences in the salt concentration, the buffer concentration, and pH. And what the authors found, as well as surfactants, and various concentrations of salt, surfactants, and buffers. And the authors found that these differences didn't prevent length measurement on mass photometry, as all of the samples were measured in the same conditions, just dilution onto the system in TE buffer. And the authors highlighted that the monomer lengths that were measured were within 3% of each other, and that the dimer lengths were within 2%. And yes, the authors highlighted how the matrix robustness and overall simplicity of sampling with MP is a large boon for in-process sampling. And then now I'm gonna hand over to Jacintha to walk you through viral vectors, applications for MP.
Thank you, Salma. Now let's look at some data sets generated for viral vectors using mass photometry. So for AAVs, the mass photometry, or SAMXMP, was used to measure the purity attributes, specifically the capsid loading. So this is a data set from a publication done by Takeda, where they measured 3 different AAV serotypes. So going from left to right, we have the AAV5, AAV8, and AAV9, measured an MP shown as the purple graph, or the purple bar, against AUC shown as the blue bar. As you can see, with each of these serotypes measured, 4 different populations were identified. So we have the empties, partially filled, full, and the overfull capsids. And one thing to highlight here is that, so the percentage for each of these populations measured were aligned between the 2 methods, which is MP and AUC, that was employed for this experiment. So this really helps us conclude that mass photometry, or SAMX specifically for AAV characterization, is serotype agnostic and can reliably measure your 4 different AAV populations, as shown on the graph here, all in alignment with the industry standard, which is very widely used in the industry, which is analytical ultracentrifugation, or AUC. So process optimization, or process development, as we know, is a very key aspect in the drug development process. And in the process optimization phase, upstream process carries higher weightage because that helps define or set the quality of the product for later stage in the process. So one thing that the industry speaks about is the lack of reliable or robust analytical tools to help measure purity profile or other attributes for AAVs in early stage of process, as in measuring crude or harvest material. So here again, this is a data set from a publication by Takeda, where they actually employed MP, mass photometry, in combination with a simple cleanup method for determination of the best or the optimal transfection mix, or the transfection condition. So in this experiment, they took the AAV9 harvest material, and a simple cleanup method that was used here was the porous capture select AAV9 affinity resin, which takes roughly about an hour or so. So they purified it using that capture resin, and then it was measured on MP, which is the darker shade of both colors on the graph on the left, and AUC, which is the lighter shade of both colors. And for this experiment, they tested 2 different transfection mixes. So transfection mix A is the green, and transfection mix B data is the red. So as you can see, you have 4 different populations identified again, using both the methods for 2 different transfection mixes. So one thing to highlight is that your partially filled population you have a significant amount of those for both of those transfection mixes, transfection mix A and B. But although that is the case, the overall purity profile is better or the best between the two for transfection mix B. That is because if you look at the full population, which is your desired capsids, because it contains your transgene of the intended length encapsulated within it. So your full population for transfection B is significantly higher compared to your transfection mix A. So therefore, here again, you're able to understand that the significance of having the right tools, the robust tools to measure the quality of your product or quality of every single factor that is optimized during every phase of process development. So this is yet another example where SAMX, or the data obtained for SAMX, aligns very reliably with the industry standard AUC. So SAMX also has the GMP software and is now adapted in the GMP manufacturing and also for QC release phases. So one thing to highlight here is that mass photometry, or SAMX specifically, has been recognized by a lot of regulatory agencies. So we have mass photometry featured in both the British Pharmacopoeia and the USP as capsid content or capsid loading under purity attribute as an analytical tool. So USP specifically, or US Pharmacopoeia specifically, they featured MP, mass photometry, for both product process characterization and also for release and stability, as shown on the left in their gene therapy analytical guide. And finally, NIFTC, which is the FDA equivalent in China, they've recently released a publication or published an article where they've highlighted the significance and reliability of mass photometry for measuring the purity attribute, again, the capsid loading or capsid content of AAV samples. So all of this together helps us conclude that SAMX is reliable as in alignment with the industry partners for measurement of the purity attribute. And because of it having the GMP software, it can now be included in both PD for process development or process optimization, and also in the GMP phase for manufacturing purposes and also for release instability. So now let's look at large viral vectors. So these include adenovirus and lentiviral vectors. But now we'll be looking at macro mass photometry specifically because these larger viral vectors are around 80 nanometer to 120 nanometer. So the macro mass photometry, or Kiritro specifically, was used to measure purity attribute for these large viral vectors. So for adenovirus, similar to AAVs, you're looking at capsid loading. And for lentiviral vectors, because of its heterogeneity, you're looking at the heterogeneous populations across process development or comparability between batches. So let's quickly touch on the basics again before we dive deeper. So Salma already spoke about how mass photometry differs from macro mass photometry. So very quickly, mass photometry measures the mass of your particles, whereas macro mass photometry gives you the mass-based contrast, but also gives you the size or the diameter of the particle, which is obtained using the Z-sweep that's employed as part of the measurement. So now with that said, you have two data segments given for every sample within the same measurement. You have the size and the mass-based contrast. So with both of those, now you're able to measure complex and heterogeneous samples, and you have a higher resolution to actually define or measure the different populations within these complex samples. So this scatter plot on the left was actually this is a measurement from a heterogeneous adenoviral sample. And as you can see, you have three populations that were identified by the Kiritro MP. So the bottom left, which is low mass or low contrast and small size particles, those are probably your fragments, capsid fragments, and also your protein aggregates. There were also two other populations that were identified. As you can see up there, you have the empty and the full populations. So they are both of the same size, but they differ based on the contrast. So your full population, which contains your gene of interest, has a higher contrast, and that's how it's distinguished on the contrast scale from the empty or partially full capsids. So now this high level of resolution, which is not present with other assays that are currently used, for instance, NTA and DLS, which don't have this contrast base, which cannot differentiate the two populations. and also NTA and DLS, which are actually impacted by the bias coming from higher particles. So because of all of that, all of those disadvantages in the current methods, Querétaro or macro mass photometry has an edge over all of those other technologies to reliably measure complex and heterogeneous samples. And this is how we could use Querétaro for process monitoring or for manufacturing purposes to try and understand how these populations or the purity of these populations are impacted across the process or during the drug development phases. So although we showed in the previous slide how Querétaro can be reliably used to measure the purity as in the empty full populations in every adenoviral sample, we wanted to understand the reliability or accuracy of the measurement across the working range. So for this experiment, we took the empties and full adenoviral samples, and we mixed them in different ratios going from 0% full to 100% full. And these were measured not only on the macro mass photometry, which is the Querétaro, but we also wanted to compare it to the other orthogonal techniques that are currently used in the industry. So when the measurements were plotted as in on a correlation fit, when we plotted the percentage full as in the theoretical calculated versus the percentage full capsids that were measured on each of these equipments, we showed that the best correlation fit was obtained for macro mass photometry as highlighted in orange, with an R square greater than 0.99. So this again highlighted that you have your Querétaro or the macro mass photometry, which can reliably measure your purity attribute for complex heterogeneous adenoviral samples, which is more accurate and reliable than other orthogonal techniques available over a very wide working range. We also wanted to understand if Querétaro can be used in other critical development workflows. And one of the key aspects, if we think about in drug development process for vaccines or gene therapies, is stability testing, because it helps you understand how the integrity of the particle or your sample is impacted by different conditions, including time, temperature, buffer components, and so many others. So here we actually tested the stability of the adenoviral sample using Querétaro by actually subjecting the sample to heat at 47 degrees over different time points. As you can see from the image on the right, we have 3 different time points going from 0 minutes to 60 minutes. So at 0 minutes at 47 degrees, you have your intact full population, which is about 50%. As the exposure to time increases, you see a depletion of the intact population going down to about 0.3% at the highest exposure time point of 60 minutes. Another thing that you notice is an increase or appearance of the degradation products in the lower contrast, lower size range. So this is very critical because this shows that Querétaro can be a stability indicating tool. Its resolution and also its sensitivity helps really pick up any subtle changes which can come either from the stability and also from other factors that's included across the process development or the drug development phase itself. So therefore, again, you're able to see, again, 2 different factors here, right? So you're able to see subtle changes. You're able to see the development or the increase in the presence of degradation products, which can be measured and quantified, and also the loss of your intact particles, and all of this within the same measurement. So for lentiviral vectors, as we touched on in the introduction slide, lentiviral vectors, as we all know it, are very heterogeneous in nature. It's really hard to understand loading versus unloading. It's a very new concept. And also, there is a huge gap in the analytical toolbox for lentiviral characterization or also for process monitoring. So here we wanted to understand if Querétaro can actually help monitor as in understand the enrichment or losses of your LVV particles going across the different unit operation stages. So this data was generated with our industry partner, where they're actually developing their LVV platform. So as you can see, going from left to right, you have the crude sample, and then the different steps in the process, which could very much be clarification going all the way down to sterile filtration. One thing I want to highlight here is that the gating strategy used here. So if you recall from the adenovirus, we had the lower box, which identified fragments and proteins. And then you had 2 boxes of the same size, which were much stringent, which showed you the empty versus full. Whereas here, again, we haven't shown you the gating strategy, but the gating strategy was developed using positive controls, as in the LVV intact particles, and negative controls, as in mock transfected or just cell supernatants, which would generate exosomes or other extracellular vesicles. So you have your lowest C gate, which is, again, low contrast, low size, which are your smaller vesicles, your cell debris, fragments, and protein aggregates. You gate B, which are your extracellular vesicles. Again, we have positive data to show those are extracellular vesicles. And then finally, your gate A, which are your LVV particles. So as you can see, Querétaro can be used very reliably for process monitoring. So here you're able to see how if you look at gate A specifically, you're able to see how there's an enrichment of LVV particles, but also in some steps losses. So at step 3, you can identify that there is a loss there. So this really helps you, because as of now, the material produced are very poor quality because we have very poorly developed processes which are not process monitored with the right analytical tools. So now you have the right analytical tool, which is quick, less complex, very easy to use, and can provide real-time data for you during the process development, which would really help you understand which buffer condition, which temperature, which pressure, all of that, which can positively or negatively impact your final material. And then finally, this is a data set generated by the Kumamoto University, where they utilized Querétaro as a pre-screening tool. So what they were looking at is trying to understand the effect of anti-HIV drugs on LVVs. So LVV is used as a surrogate here for HIV in the screening study. So as you can see from the scatter plot on the left, you have wild-type LVV, and they also used mutant, which is the drug-resistant LVV. So one thing to highlight here is you see a significant amount of LVV populations in your gate A. When the wild type and the mutant treated with the anti-HIV drug, the mutant is resistant, whereas your wild type, you see a significant drop in the population, which is more evident as seen in the bar chart on the far right. So as you can see, you have the wild type, which is not treated, wild type treated, mutant not treated, and mutant treated. And one thing that really is significant there is your wild type, which is treated, which has a significant loss of particles. So we've shown here enough evidence that Querétaro or the Macro Mass Photometry can be used as a purity measurement for complex samples and heterogeneous samples like adenoviruses and lentiviruses, both in the vaccine field and also in the gene therapy field. So to wrap all of this up, yes, so we've shown enough evidence again that mass photometry, which also includes macro mass photometry, has provided good data for critical quality attributes for the different applications discussed above. But again, yes, there are other tools out there that can do the same or something similar. But we want to highlight here that as in why mass photometry stands out in comparison to its orthogonal techniques. So mass photometry or macro mass photometry, they have very minimal method development. And one thing to highlight here is that they can be adapted or adopted across modalities like how we've shown here, going from mAbs all the way to large viral vectors. Another thing is, irrespective of the modality used, sample preparation is very expensive, which means whichever technique has low sample demands for measurement is key. So unlike other tools which need high micrograms or high microliters or milliliters of samples for measurement in analytics, here mass photometry only requires 5 microliters. And then also we have near real-time analysis, which we spoke about a lot during the presentation, which really helps with either drug development and specifically during process monitoring phases, because during the development phases, you want real-time measurements, which can really help make the decisions quicker and move on to the next unit operation. So with mass photometry, you're able to get your measurements in less than 1 minute. And with the automated acquisition, you're able to measure either 24 samples or 14 samples in 90 minutes with either mass photometry or macro mass photometry, respectively. And finally, as Salma described the principle and the working operation of how mass photometry or macro mass photometry works, you would have understood now that it's very ease of use, it's very user-friendly. We have a very intuitive software and a very intuitive data analysis, and it's a very small and a benchtop equipment. So all of that put together, you can really have the training to a new personnel done in less than a day. So all of this put together should have helped convince that mass photometry really is reliable, it's accurate, it's sensitive, and can be adapted for different critical quality attributes for the applications talked about above. So to quickly wrap up, but also introduce our different instruments in our portfolio. So we have two mass photometers. We have the 2MP and the SAMX MP. Both of it have the manual and the automated versions of it. So they cover different mass ranges as seen on the right. So 2MP has demonstrated good use in proteins, antibodies, and the nucleic acid space, whereas SAMX MP was specifically designed for characterization of AAVs. And as mentioned previously, the SAMX MP has the GMP software, so it can be adapted in both the PD and the GMP environments. And the newest inclusion is the Curator MP, or called the Macro Mass Photometer, which is designed to target larger viral vectors such as adenovirus and LVVs in the size range of 40 to 150 nanometer. So to finally end with our journey, so across the 7 years that Refine has been functioning, although it was a new technology, the mass photometry, because of the reliability and the significance of it in different applications, we've had worldwide adoption. And two things to really highlight here is that we have over 1,000+ publications, which really highlight the use of mass photometry, again, in the different modality spaces. And also in all of the adoptions of more than 500 instruments, in about 20 biopharma companies, we've actually been successful in adoption of mass photometry or macro mass photometry in about 18 of them. Thank you for listening in, and we're happy to take any questions.
Thank you for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Q&A box and click submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started with our first question here. This question is asking, what unique insights does simultaneous mass and size measurement, or MMP, provide that MP alone cannot?
So with mass photometry, like we saw in the presentation, you're looking at just mass-based measurements. And obviously with macro mass photometry, you have the two data segmentation, which is the size and the mass-based contrast. So for some like larger viral vectors or non-viral vectors, where these are larger particles, especially if these populations are heterogeneous and there are populations that fall within coincidentally the same mass range, your MP wouldn't really have that effect to show you the distinguishing between the populations. So that is where macro mass photometry comes in, where for these viral vectors, you also would like to understand the size of the particles to understand the integrity, right? And also, if you have these heterogeneous populations falling within the same mass range, you're able to distinguish them now because you have the size measurement. So overall, if you think about it, your macro mass photometry fills that gap that mass photometry cannot, especially, like I said, for larger particles where you can have heterogeneous populations falling within the same mass range. So you're looking at, like with the macro mass photometry, you're really looking at the integrity of your particles. You're able to differentiate between empty folds like in adenos, or also for heterogeneous populations like we saw for LVVs. So overall, it's giving you an insight of the overall quality of the product or purity of the product.
Excellent. Thank you so much for that. Okay, our next question that we have here is asking, how do MP and MMP simplify analytical workflows compared to traditional techniques?
That's a great question, Cassie. Let me take this one. I think there's probably two ways that mass photometry technologies are helping you out for simplifying your analytical workflows. The first is probably the best part of mass photometry. It is so easy to do, so anyone can pick it up and anyone can run a measurement without the need for a subject matter expert. Anyone can pick up how to use a mass photometer in about less than half a day of training. A fun thing is we tried this out with our finance team, where we got some of them to run their very first biological measurements on a mass photometer after half a day of training. So we've tested this out in-house. And then on the second front, I would say it's probably just in terms of how easy the methodology is. It's mass photometry measurements involve one universal dilution step. So this means you can avoid having to deal with some of those cumbersome method development steps you have to deal with like HPLC methods, where you have to do column optimization or buffer optimization. And those are probably my two top points where it would simplify your workflows.
Excellent, thank you. Okay, moving on to our next question here. This next question is asking, what are the sample volume and cost benefits of using MP or MMP at early development stages?
I think I will take this one again. I would say, so let's think about what are the sample volume and cost needs of a mass photometry tech measurement, either for mass photometry or macro mass photometry. You're using about 5 to 10 microliters of sample. A measurement costs less than $10, and it's taking a minute to acquire a movie. So that, and also that 5 to 10 microliters of sample you're using is in the nanomolar concentration range. So that's going to be nanograms of sample. So if we compare this to traditional technologies like HPLC methods, that's going to be about 15 to 20 times faster, using around 100 times less sample. Or other technologies like AUC, where that's going to be like a 6-hour run is going to be, I think it was around at least 100 times faster. And you're using hundreds of micrograms of sample, so it's 1,000 times faster for 1,000 times less sample is going on a mass photometer. So that has a lot of value when you're working earlier in development, say in discovery or earlier development process stages where you are limited with your sample amounts. So being able to use these low sample volumes and low concentrations, just minimal sample amounts of nanograms of sample, helps you get information on your samples earlier, so you can inform your processes for downstream sooner.
Excellent, thank you. Okay, and we have a ton of questions coming in. That's wonderful. Please keep submitting questions. If we don't have time during the live, we will email you privately. Our next question here is asking, how do these methods reduce hands-on time and enable walkaway operation?
This is another great question, Cassie. We didn't have a chance to go over in detail the different products that we have available. So we've discussed the different product lines where we have the 2MP for that 30 kilodalton to 5 megadalton mass range, the SAMX line for that's optimized for AAVs that fall up to around 6 megadaltons, and then the Caritra line for those larger larger viral particles. But what we didn't mention is that our 2MP line and our SAMX line also have automation available there. So that's essentially taking that benchtop instrument, that mass photometer, sticking it inside a pipette robot so that you get the robot to do all your pipetting for you. So that's another step less to deal with in your mass photometry workflows. So in that situation, when you have the automated version of the 2MP or the SAMX, you're able to run to automate acquisition of 24 samples in 90 minutes. And then our Caritra product line for the macro mass photometry measurements that you'd want to do with larger particles is running 14 samples in about the same time, in about 90 minutes as well. So that enables an added level of walkaway. So you don't need to be standing at the instrument for all of your measurements to be taken.
Amazing. Thank you. Okay, moving on here. Our next question is asking, what is the value of using MP or MMP data for go, no-go decisions in drug development pipelines?
Yeah, I can take that. So it's a really key aspect. The question's great because that's one of the key aspects or key discussions happening in the industry right now. So one gap right now in the industry that everyone is recognizing and working towards is that there aren't robust or reliable tools that can be used early in the process. So if you look specifically for viral vectors like lentivirus or AAVs, people just use ELISA PCRs, take a ratio, which really causes a misinterpretation on the quality of the product. But people have been using it mainly because the other tools that are available, which are reliable methods, either complex, expensive, or outsourced, right? So we have been having issues with the quality of the product because you start off in the upstream with par quality, and then obviously later stage in the process, now you have issues with poor quality. So you have to have delayed timelines because you're trying to rework it and also increase costs, right? So again, that's a very significant part in upstream, right? So now that we have MP and MMP, so MP will use it in combination with a simple cleanup method, like I spoke about. There is another webinar if you're interested in our website, which talks about all of the different methods that can be used. But very briefly, these are all column-based methods, monoliths, resins, or even beads that can be used to quickly purify and then run it on the MP. MMP can just be run with the crude sample, it just works. So both of this really gives you the true purity profile of your product through the upstream optimization phase. So now you have reliable data and you're making your decisions in upstream, which is data driven, right? So now you're able to say that, okay, this is par quality data, we will not go through it, or this sample or this material is of higher quality. So you're able to proceed and get overall high quality at later stage in the process. So yeah, overall, if you think about it, MP and MMP provides very high value, which current tools do not provide, and can really help fill the gap there is in the industry right now.
Excellent, thank you. Okay. Our next question here is asking, can MP or MMP be integrated into QC release in GMP environments? And if so, how?
Again, a great question. I'll take that because it falls in my remit. Sorry. So as we spoke about SAMX specifically, so we have 3 in our portfolio. Yeah, 2MP, SAMX MP, and Kevitro MP. So the SAMX MP has the GMP software, and it has been adopted by a lot of big pharmas and other companies in the GMP environment. They validated it and really using it for both process monitoring during manufacturing and also for QC release. But one of the key aspects, again, it's part of the presentation, is regulatory recognition. Because if you think about it, this is a new technology, and the regulators really need to understand them, accept it, and also help define the analytical toolbox for the specific therapies. So again, like in the presentation, you have the USP, US Pharmacopoeia, which has really featured us very well via in their gene therapy analytical guide as a capsid content method. We are also highlighted in their general chapter 1067, where they really outline the manufacturing and QC for AAVs. And we are again in there as process and product characterization. We have the British Pharmacopoeia highlighting us. And finally, the NIFTC. So overall, all of this really tells you or gives you the confidence that mass photometry really can be integrated. There has been, a data to show that it's validated, can be integrated in GMP environment, both for process monitoring and for release. One thing I would like to highlight here, so the MMP, which is the Kevitro, and the 2MP, which is MP, mass photometry, do not have GMP softwares at this point, but we are working to get that developed as well. So we've showcased that MP, which is SAMX MP, can be in the GMP space. So very soon we'll have the 2MP and Kevitro in that space as well.
Okay, wonderful. And it looks like that is all of the time that we have for questions today. So I want to thank you both for your time today and your important research. We would also like to thank LabRoots and our sponsor, Refine, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted during the live and the on-demand period that we did not have time for will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We highly encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.